Hi everyone, this is Dr. Madhuri from Team MDSCONQUER. So now I'm going to discuss about the sterilization and the disinfection in this video, which is very important in the present scenario. So you have to make a note of all these points. So these are the contents of the video. So this is the disinfection and sterilization. Disinfection includes, it is a process of eliminating the most harmful microorganisms, okay? Whereas the sterilization, it includes like it will kill the microorganisms and also the resistant bacterial spores. This disinfection can be used only on the inanimate substances or the surfaces. So disinfection process includes two steps. You can make a note, that is, first step will be of your vigorous scrubbing and the second step will be of wetting the surface with a disinfectant. Then there are two techniques. One, the technique is spray wipe spray technique, and the second technique is wipe and discard wipe. So here the spray wipe spray technique includes the mechanical cleansing with a paper towel, then followed by a chemical action of the disinfectant solution. Whereas wipe discard wipe technique includes first wipe remove the microbes and the second wipe will provide the disinfectant. So these are the sterilant level of disinfectants given in this table, which is very important. So if you take the sterilant, it will destroy all the microorganisms, and these are the examples and where you have to use the sterilant. And the high level disinfectant, its definition, examples and the usage. Whereas intermediate level disinfectants, its definition, examples and the usage. Whereas the last thing will be of your low level disinfectant, what is the definition and example, and this is the examples where you have to use, and this is the usage of this. And these are the disinfectants, type of disinfectants you'll use in the daily practice. This is for the alcohols. These are the examples include, and the mode of the action will be mainly by the denaturation of the proteins, and these are the advantages and disadvantages of the alcohols where you have to mention your exam. Then coming to the phenols, that will work by the precipitation of the proteins, and these are the advantages and disadvantages of the phenols. And aldehydes, mode of action is denaturation of proteins and nucleic acids, advantages and disadvantages. And for the chlorides, these are the interferes with the cellular metabolism. It has an oxidative action, and it has a lipid and fatty acid degeneration. These are the advantages, and these are the disadvantages. Then coming to iodophores, it will disrupt the nucleic acid and protein structure. These are the advantages, and these are the disadvantages. Quaternary ammonium compounds, it will impair the bacterial cell wall permeability, advantages, disadvantages, and these are the examples. Then coming to the sterilization, this is the physical agents of sterilization, which include sunlight, drying, moist, heat, filtration, radiation, and ultrasonic and sonic vibrations. And this is the chemical agents which are used. So these are the steps in the decontamination process. First is the cleaning, then removal of debris, then the manual cleaning, automated cleaning, or environmental protection agency is used. So the instruments, how to disinfect is, first step will be of your pre-soaking. It will prevent the drying and saliva of the blood. Then one should place loose instruments in a perforated cleaning basket and place in the holding solution. This is the pre-soaking. So in dentistry, there are four accepted methods of sterilization, that is autoclave, chemiclave, dry clave, and ethylene oxide sterilization. If you take the dry heat sterilizer will transfer the heat energy to the instruments, and mode of action will be of your protein denaturation, oxidative damage, and toxic effects or elevated level of electrolytes. And these are the types: static air at 160 degrees Celsius for 2 hours, and forced air type for 190 degrees Celsius for 6 to 12 minutes. So these are the instruments which can be used there. And in the moist heat sterilization, these are the steps. And this will saturate the steam, comes in the contact with the cooler and condenses the water and gives up the latent heat to the surface. So there will be a large reduction in volume, sucks in more steam to the area. This is the process of moist heat sterilization. Then the types are of down displacement or a gravity steam sterilizer or a vacuum steam sterilizer. And coming to the sterilization cycles, this is the standard cycle, and for the fast cycles, these two important things you have to mention. Then there will be of results are consistently reliable, good penetration, time efficient, but the disadvantages includes blunting and corrosion of sharp instruments and damage to certain rubber goods. Then coming to the unsaturated chemical vapor sterilization, there are of Harvey sterilizer or a chemiclave, and where the 0.23% of formaldehyde is active, and 72.38% of ethanol, acetone, ketone, water and other alcohols are used. Advantages of this thing and disadvantages. Coming to the ethanol oxide sterilization, it is a flammable. So the mechanism of action will be for your alkalization of the protein, DNA and the RNA. Okay. Then this gas concentration, this the temperature used and the exposure time mentioned. Then coming to the glass bead sterilization, temperature mentioned here maintained will be of for 220 degrees Celsius and time will be of 10 seconds, mostly used in your endodontic files, burrs and rotary instruments. Then with the radiation will be of two types, ionizing and non-ionizing radiation. So this is the ionizing radiation, and this will be of called as a cold sterilization and act by the free radicals, which includes gamma rays, X-rays and high-speed electrons. Disadvantage thing is they are potentially dangerous to the human cells. Then non-ionizing radiation, mode of action will be distraction of nucleic acid through induction of thymine dimers, used for the operating rooms, isolating rooms and biologic safety cabinets. Disadvantages will be your poor penetrating energy. Then instrument reprocessing. So those are things about the infection and disinfection and the sterilization of dental instruments. Then coming to instrument reprocessing. So this instrument reprocessing is the starting with the cleaning, then disinfectant, inspection, packaging, sterilization, transport, storage, use, again transport for cleaning. So this is the cycle where this thing is processed, instruments are processed. So these are the two steps, that is cleaning and the packaging. Then after that, sterilization, storage. So those are the important steps you have to mention with the reprocessing of your instruments. Coming to the waste management, which is very important. So management of biomedical waste will be of your general office waste or a biomedical waste. Okay, biomedical waste again includes if any anatomical waste, non-anatomical waste or a plastic waste. So these things, this color mentioning code are very, very important. You have to mention in your examination if they give an infection control or a sterilization question. So this black is for the cytotoxic drug and chemical waste, red is for the soil waste, yellow is for anatomical waste, blue is for infected plastic, and sharps for the needles and the cut glasses. Even you can color your thing with the light colored things. Okay. So these things you have to mention your exam. And finally, these are the references of this video or the topic. So I think you are clear how to mention or what to write if an infection control, sterilization, disinfection is given for you.